A single modern clinical trial generates millions, sometimes billions, of individual data points. Before a new therapy reaches the market, it produces a silent accumulation of information, like lab results, digital imaging scans, and continuous telemetry from wearable devices. Behind every number in that flow is a patient. A shift in a biomarker or a missed dosage log represents a real medical outcome, determining whether an experimental treatment is effective or toxic. For decades, managing this information relied on paper case report forms. Research coordinators transcribed patient charts by hand, shipping physical boxes of paper to central facilities, where operators keyed the entries into basic databases. That system reaches its limit with modern trial designs. When you introduce remote patient monitoring and adaptive protocols that adjust dosages based on incoming results, the data volume scales beyond the processing capacity of human reviewers. Relying on manual reconciliation for millions of entries introduces transcription errors and creates bottlenecks. At this scale, human cognition alone cannot verify the accuracy of the data without compromising the study's timeline and integrity. The gatekeepers of modern medicine, regulatory bodies like the FDA and the EMA, require absolute proof of safety and efficacy. They demand to see the origin and audit history of every single observation made during a study. A handful of missing lab values or undocumented corrections can cast doubt on an entire study. If inspectors find discrepancies between the source documents and the final database, years of research can be invalidated, pausing the drug's approval process. To survive this level of scrutiny, the industry moved away from manual transcription. They required a system that could verify data integrity from the moment a physician logged a symptom to the final statistical analysis. This shift moved data collection onto high-speed digital infrastructure. Investigators began entering clinical observations directly into digital systems at the trial sites, creating an electronic record at the point of care. Raw observations now pass through a multi-layered verification process. Only data that survives this continuous digital review is used as evidence for regulatory submissions. The first layer of defense is the Electronic Data Capture, or EDC, system. This is the centralized digital environment where clinical trial data is recorded, standardized, and secured. When analyzed, the laboratory system encrypts the blood results and transmits them directly into the EDC. The data flows from the testing instrument into the central database without the need for manual data entry. The EDC subjects the entry to the Alcoa principles. It verifies that the data is attributable to a specific source, legible, contemporaneously recorded, original, and accurate. Every action taken on a record requires an electronic signature. The system generates a cryptographic audit trail that logs the timestamp, the user ID, and the reason for any change, ensuring that every modification is traceable and authorized. This process converts individual observations into a permanent record that withstands regulatory scrutiny. By securing the origin of each data point, the EDC creates a legally binding account of the trial's findings. A structurally secure database can still harbor medical inconsistencies. A secure entry logging a mild adverse event might contradict a free text physician note describing symptoms that required immediate hospitalization. To resolve these nuances, clinical data managers integrate large language models into the review workflow. These AI systems read and cross-reference unstructured narratives across the entire patient history. These algorithms apply clinical heuristics to detect contradictions. They can instantly flag if a sudden drop in hemoglobin levels is inconsistent with a patient's medical history, a task that typically requires weeks of manual human review. This chart shows the throughput of clinical trial data cleaning, comparing traditional spreadsheet methods against the AI-assisted Octosi platform. The results demonstrate a significant acceleration, with reviewers experiencing a 6-fold to 15-fold increase in processing speed. This scatter plot maps the relationship between speed and accuracy for individual reviewers. Even as processing volume increases, reviewers using the AI platform move toward near-perfect accuracy, outperforming the highest-performing spreadsheet users. These confusion matrices compare discrepancy detection. The AI system reduces false positives 15-fold, distinguishing anomalies from expected variations. The metrics table quantifies this. The manual error rate dropped from 54.7% to 8.5%. Mistakes occurred in fewer than 1 in 11 data points. This shifts the cleaning process from a manual review of past errors to a real-time assessment of data quality. AI allows reviewers to focus on complex medical decisions rather than repetitive data reconciliation. After the data survives the EDC and AI verification layers, all queries are resolved. The biostatisticians and medical monitors complete their final reviews to ensure the database accurately reflects the trial's events. This leads to the database lock. This is the final regulatory milestone that signals the data is ready for formal reporting and analysis. Once locked, the database is effectively immutable. Write access is disabled, and no further changes can be made without an official, documented unlock process. This locked dataset is the only version used for the clinical study report and regulatory submissions. It ensures that the final results are based on verified, unalterable findings. The combination of automated capture and AI review shortens the total clinical trial timeline. Historically, the window between the last patient's visit and the database lock could take several months. With these integrated systems, that period is reduced to just a few weeks. Reducing this delay saves millions of dollars in operational costs per trial. It also lessens the administrative burden on clinical sites, allowing research teams to focus more on patient oversight than data queries. In clinical research, operational speed directly translates to how quickly evidence can be analyzed. Compressing the data cleaning phase allows for faster decision-making by both sponsors and regulators. In trials for aggressive cancers or rare diseases, these operational efficiencies can shave critical months off the total development timeline. Saving weeks during the cleaning phase directly shortens the path to regulatory review. The integrity of this digital gauntlet ensures that the final results are credible. When a treatment is approved, the public can verify that the safety and efficacy data were protected from the moment they were recorded. The industry has moved away from the risks of paper transcription and toward a framework of automated checks and auditable records. Computers and AI have standardized the path to regulatory approval. These tools changed how data is managed, turning raw observations into actionable, life-saving information.